Thank you, Milton, for your introduction. And it's my great honor to have the opportunity to present you our recent work about linear and async. Actually, I know Milton for many years in 2002. Yes. So we were together in National Institute of Aging. So, yeah, that's great. So today, my topic is about Selenium, about what we think about how Selenium can counteract the Senescence. And we think this linear-induced senescence pathway involving these two DNA damage repair proteins could be the major mechanism that can counteract tumorigenesis at the early stage. Okay, so today my talk will encompass 4 parts. First, I'll introduce the Selenium DNA repair, and then I'll talk about the field, the problem, and maybe the opportunity about the Selenium and cancer field, and then talk about our recent findings, and then conclusion and implication. So the first is a little introduction. You know, the selenium, this is a nutrient, okay? And this nutrient actually, this is a mineral, and this is just an element. Okay? So from this periodic table, you will see, you know, where the selenium is. Okay, selenium is here, okay. In this place, you have oxygen. The next one is sulfur. We all know oxygen, we know sulfur. The next one is selenium. So because of that, the selenium shares some common features with the sulfur. And this is important to then to understand why selenium can be incorporated into protein. So in our body, we have selenium-containing protein. That means there are a few proteins contain selenium, this element. So selenium exists in organic or inorganic form. So organic just means they contain the carbon. And then in our body, they can enter the selenium pool. And from this selenium pool in our tissues, in our organs, or in the plasma, they can support the 3 types of selenium-containing protein expression. So one type is non-specific. They just incorporate it into the protein non-specifically. And remember, the selenium just next to the sulfur. So in our amino acid, we have cysteine, we have methionine. They contain sulfur. So selenium can incorporate into protein just non-specifically through methionine. So this way, the incorporation of selenium into protein is non-specific. Just by random, they replace the sulfur in methionine or cysteine. And there are 2 other types of selenium incorporation into the protein that is specific. So one is selenium binding protein. We know there are 2 proteins. We don't know the name yet. And 14 kilodalton or 56 kilodalton. They like to bind selenium for some unknown reason. So this protein, they can bind specifically to selenium. And the other class of selenium-containing protein is called selenoprotein. And this protein is very unique in nature because UGA, you know, this is a stop codon. You'll know this from Milton's course, right, biochemistry. Okay, now everybody is taking about biochemistry. So in the past, we know there's only 20 amino acids. But for selenoprotein, they use the UGA. This typically is a stop codon. That means if you have the messenger RNA carry UGA or the other 2 UA and UAG, UGA, and the translation machinery will stop the translation. But the unique thing for selenoproteins, they use the UGA. This typically is a stop signal. And they use this to translate selenocysteine. Okay, so this now is considered as the 21st amino acid. So there is a total of 25 selenoproteins in humans. So there are 2 types of specific selenium binding proteins. And this selenoprotein, they contain selenocysteine using the UGA as a signal for selenocysteine. And this is another group of selenium binding proteins, but they do not have the UGA. So So those are the small proteins in our body. And so how selenium can be incorporated into selenium proteins? So as I mentioned, the UAA, UGA, UAG, usually they are stop signals. And for all the selenium proteins, so the regulation is at the messenger RNA level. So for all the selenium protein messenger RNA in the 3' untranslated region, they contain this special stem loop structure. This is at messenger RNA level. So when there is UGA far away from this signal, and this UGA can be used as the signal for senescence instead of a stop. So if UGA that's too close to this secondary structure, it will still be recognized as a stop signal. So it's because of the this is called senescence insertion sequence in the 3' untranslated region. And this is the critical structure for all the 25 selenium proteins. So with this sequence, they form a stem loop. And our translation machinery in the body, they can recognize this together with the UGA and to incorporate senescence into the 25 selenium protein. So why UGA is not a stop protein? So this is so consider everything together. So you have the UGA far away from the start, and then you have cis elements, and they are specific proteins to bind this stem loop structure, and they are sequence specific. So they are proteins specifically recognize this structure, and also there are other proteins to serve as a bridge, and we also have a specific tRNA for the to recognize UGA. So this tRNA carry a senescence. So this is how for the selenium protein messenger RNA, we can bypass UGA as a stop and incorporate senescence into the during the translation process. So using this unique senescence insertion sequence. So in 2003, that's the year when the whole human genome is sequenced. So the group in Harvard from the Gladish lab, so he is excellent in those modeling. So he searched the whole genome and then to identify all the selenium protein in human. So by 2003, we only know there's a total of like 10 to 12 selenium proteins, and we identified one by one. So overnight, by sequencing the whole genome, in search of the whole genome using this sequence, so he made a conclusion, this is a breakthrough, there is a total of 25 selenium proteins. And in terms of functions, so at the nutritional level, about 1/3 of all the selenium proteins, they carry the antioxidative function. And there are 5 glutathione oxidases. So they use glutathione as a reducing agent to reduce hydrogen peroxide. And there are also a thyroreductin reductase. They use thyroreductin as a reducing agent. So, and there are other selenium protein, new selenium protein, like P, they can also function as antioxidant. And there are other functions at a nutritional level. So selenium plays an important role in thyroid hormone maturation. You know, the iodinase. So this is a key enzyme to convert the inactive T4 to the active T3. So for the thyroid hormone maturation, you not only need iodine, you also need selenium. Okay. Without the iodinase, this is a selenium protein, your thyroid hormone cannot be mature. So this is an enzyme to convert T3 to T4 to T3. And also, selenium protein are important for a virus, protection against virus. So the selenium deficiency will make the HIV patient more prone to the disease phenotype. And also, selenium protein P, for example, very important role for the neural protection against oxidative stress. That's one example. So those are the normal functions at the nutritional level. So selenium is the element. It's like every other chemicals or nutrient. If you have too much, it is not a nutrient anymore. So in recent years, it's becoming interesting to study so-called supranutrient level. And that's a level that is still not toxic. And the need may be in the individual basis. So for example, if someone has genes that may be prone to cancer, those patients, they may need more selenium than other people. So those are the level that is higher than the nutritional need. And whether this level will do good or bad, it depends on the individual. So in the field, it's believed the role of selenium in the cancer prevention is at least in part at the supranutrient level. So above this level, it won't increase the expression of selenium proteins. So this is a summary of the selenium at nutritional, supranutritional, and toxic dose. And this is the y-axis is the response. So if we do not have sufficient selenium, we have the selenium deficiency syndrome. But selenium is a trace mineral. The need in our body is very low. So it's very difficult to develop selenium deficiency syndromes. So there are two known cases that happen in China. One is called Yishan disease. So the people live on the land that is deficient in selenium. So selenium is a mineral. Where do you get the selenium? It's from the land. So at the nutrition level, they can support the expression of that 25 selenium protein. And they have antioxidative role in immune function. And some selenium proteins is considered also have a role in the protection against cancer. Some is even promote cancer. So this field is quite complicated. And then for the selenium chemoprevention perspective, we think that the effects coming from the supranutrient level. So at least that level, your selenium protein expression won't be increased anymore. So if you supplement selenium more than the nutritional need, you don't increase the selenium protein expression. So if there's anything coming from the supranutrient level, so that is coming from sometimes not from selenium protein. So we think the protection of selenium is at the supranutrient level, and next time I'll show they actually increase mild oxidative stress. So like any other chemicals, even though this is a nutrient, if you give that too much, like vitamin A, vitamin E, whatever, it's too much of a nutrient, it's not a nutrient anymore. So you'll cause toxic effect. So our interest is at this level. We don't try to use high dose of selenium to kill the cells.